Good morning and welcome, or actually hello, because for some people it's certainly not morning. Welcome to CyVerse Learning Institute's Focus Forum webinar. I'm Tina Lee, CyVerse User Engagement Officer. Today we're honored to host this webinar on metagenomic analysis using NanoDJ notebooks in CyVerse by Héctor Rodriguez Perez, a doctoral student at Hospital Nuestra Señora de Candelaria in Spain. He's a co-creator of NanoDJ. And for those who missed the last webinar, his other co-creator had presented on doing genome assembly using NanoDJ notebooks. For those unfamiliar with CyVerse, we're an NSF-funded cyber infrastructure project, and this free webinar series is designed to fulfill part of our mission to train scientists like you on how to use CyVerse's computational resources. So let's quickly take care of some housekeeping and then we'll start the webinar. Today's presentation is roughly 30 minutes with time for question and answers at the end. Please open the Zoom chat window and type your questions there for Hector. Materials from today's webinar, such as his presentation slides and a video recording of the webinar, will be posted on a wiki page for which we'll email you the link in a short bit. The CyVerse Learning Institute will be offering our second Foundational Open Science Skills Camp and our third Container Camp. Both are scheduled for spring 2020, so please visit our website to register and for more information. Also, please join us in 2 weeks for our next webinar by Megan Balk of the U of A entitled Git for Mere Mortals. Git is a great and necessary tool for any data scientist's computational toolbox, so please join us for that. And without further ado, I'd like to welcome Hector. He is what data scientists call a unicorn, a mythical animal who has expertise not only in computer science, but has abundant domain science knowledge, such that he could write something like NanoDJ to be used in genomics. Anyway, Hector, please introduce yourself. And my first question for you, actually, before you get started is, what does the DJ part of NanoDJ mean?
Hello, everyone, and welcome to this webinar. This is the continuation of the NanoDJ and CyVerse series. It started with the introductory webinar by Upendra, and the last day my colleague Tamara talked about genome assembly pipelines that we have included in NanoDJ. So the D stands for Docker and the J stands for Jupyter. That are basically the two main technologies that have made possible that we now have this tool called NanoDJ. I actually have a tech background, as you said. I studied computer science and software engineering, and now I am into this amazing field that it is bioinformatics. Today we're going to talk about metagenomic analysis using NanoDJ notebooks in CyVerse. Throughout this webinar, we are giving an introduction to metagenomics and its applications, and then we are going to perform different analyses with two different methodologies. First of all, we are going to build a custom database using the BLAST classification tool, and then we are going to perform a rapid taxonomic classification using Kraken 2. So NanoDJ is an integration of bioinformatic tools for nanopore data analysis. It is built on top of the Jupyter Lab environment, and it is all possible thanks to a Docker container, which is a virtual environment that can actually be run on different machines. So there is no need to install all the software and dependencies separately. We started working with the nanopore MinION about 2 years ago. And after the first evaluations that we made, we decided to build a tool for interactive and easy learning. So everything from the tools and the notebooks are all included in NanoDJ. So it is easy for new users to keep up the pace with all the software packages updates, and all the steps can be followed cell by cell, as we are doing here today. All the materials are also included in VICE, which is the virtual environment by CyVerse. Today we're using two different notebooks. They are written in the Python programming language, along with some data science and bioinformatic tools, and some command line tools as well, such as BLAST or Kraken 2. We also have two different example sequence sets. One of them is from mitochondrial DNA of different animals and plant species, and then also a 16S rRNA gene sequences of a saliva sample. Note that these both data sets correspond to real minion runs that were done in our lab. Metagenomics is the study of the genetic material recovered directly from environmental samples, like soil, ocean, or even the human body. So the DNA in metagenomic studies doesn't belong to one specific organism, but it belongs to all the species present in the sample. There are two main approaches to do metagenomics. In shotgun metagenomics, all genes in all organisms are sequenced, so it is possible to evaluate diversity, study abundance profiles, or even assemble the genomes of some of these species. On targeted metagenomics, only specific amplicons or genes are sequenced. So we use lab protocols to isolate or enrich these fragments of interest, such as the 16S gene, which is very useful to identify, for example, bacteria present in a sample. Metagenomics has a broad range of applications. It enables species discovery and diversity studies in ecology and agriculture, and also gut microbe characterization and infectious disease diagnosis in medicine. Here, we are doing metagenomics using long reads. This is actually a cost-efficient approach to do metagenomics, and it is useful to get some insights without the need of other expensive high-throughput technologies for DNA sequencing. The rapid library preparation and the real-time data output enables faster analysis and results, which is pretty unique to this technology, and this is a positive thing. Also, the long reads enable the sequencing of these informative genes, such as the 16S rRNA gene, in their entirety. So this means that we could have the full 16S gene on just one read. The first notebook that we are going to use is the educational notebook that is already included in NanoDJ. Here we're making an interactive quality control of our input file. Then we're going to build a custom BLAST database with reference genomes. Then we will perform the BLAST classification, then some post-processing, and finally the visualization of our data. So here we are. This is the first notebook, the educational notebook. This actually has some information about nanopore sequencing, the base coding process, and also some information about the sequence file formats, the FASTA and FASTQ files. In this first Python cell, we are using BioPython, which is a library for computational biology and bioinformatics. Here you could use R as well. Since I have a tech background, I feel more comfortable doing it on Python, but it is okay to do it on R. You can use the bioconductor package. What we are doing here is loading our example sequence set using this function into a pandas data frame. So the positive thing of doing that is that we can get a summary table like this. And we can, okay, so we can have the mean read length, or we can check how long is our longest read or our shortest read. Another thing we can do with this data frame is to plot the distributions of some metrics such as read length, the GC content, and also the average quality. So now we are not doing any filter. We are going to jump directly to the classification task itself. So the first thing here, we should build our database first. Here we're using the make BLAST DB command that is included on the BLAST software package. This takes the reference sequence file as an argument. And since we only have this time 210 reads, they all belong to 7 different genomes, both the database building step and the classification will be very fast. Once we have built our database, we can start with the classification. Here we use the BLAST10 tool, which is the tool made for comparison of nucleotide sequences like DNA. This takes our reads as an input, the database, And also here we are using the out format argument because Blast software outputs are really complex report files. So here we are just basically telling the program to just output a CSV file with some basic metrics. So once we have done the classification, we can take this output file and load it again as a pandas data frame. So we have this table of 3 columns with subject, the read count, and the proportion. So now I'm going to take this first column and the third and use the matplotlib library to generate this circular plot. And we should be done with this notebook from the input files to the visualizations. So now we are going to perform a rapid taxonomic classification using this long read data. So why is rapid taxonomic classification of long reads so challenging? This is because these reads are way different from the short read technologies, not only in terms of read length, but also in terms of quality. The nanopore reads have a higher error ratio, and also the databases, because the databases have been increasing in size this last year, and it will be unfeasible in time and space to try to analyze a next generation sequencing dataset with a large database and the BLAST tools. And also these databases are not perfect. I am going to speak about that later, but they are not as specific on accuracy and species diversity representation. So yes. This notebook is pretty similar to the previous one, but in this case, we have a way bigger data set to analyze. We have nearly 40,000 reads. And also the database, we are not building the database from local file this time. We are downloading a database index. And then we will perform, as the previous notebook, a Kraken-2 rapid taxonomic classification, then again some post-processing, and the visualizations. Okay, so here we are. These are actually the same Python cells as the previous notebook. I am loading my sequence file again into a data frame. And now I am plotting again the distributions, but now we are going to perform some filters, for example, of the read length. So since we are analyzing a 16S rRNA sequence file, this gene is about 1.5 kilobases long. So any read that is out of our range of 1400 and 1700 should be out of interest. So here note that the axis, the y-axis on here, it is logarithmic. So we can see there are some reads that are out of range. So we should filter them using, again, a Python function. We're going to do a filter with the average quality. Again, note that the nanopore sequencing software, it does perform a quality filter, but now we're going to be more conservative and set a higher threshold. I put it here in green, the nanopore software threshold, and here in red is our threshold. So now we generate a FASTQ file with our filtered reads. We generate a new file that is the file that we are going to use in the classification task. We have nearly 20,000 reads that have passed our quality control. So this is very nice. Now again, the database building step. The Kraken-2 includes the Kraken-2 build command. You can download any NCBI RefSeq collection. You can build standard databases. But in this case, since we are using 16S genes, it would be inefficient to use a database which contains the full sequence of the genome. So luckily for us, the Kraken-2 build command lets you download some special databases like the RDP database that we are using here. This is already built on the NanoDJ container. It takes a couple of minutes to build. Once we have built our database, it is a good practice to check it out. So we are going to use the Kraken-2 inspect command, select our new database, Let's output the first 50 lines. And here we can see we have more than 3,000 taxonomy nodes. We have some nodes at genus level, family level, order. Okay, this database doesn't get up to species, but it's up to genus, and this is okay. Now we're going to perform the taxonomic classification. We choose the database and the two output files. One of the output files is a log file, which is a little bit difficult to analyze, but you can also get a report file on here, which is the file we're going to analyze. And this is the output of the Kraken 2. Here you will see that the information, it is organized by taxonomy. We have some results at order, family, genus again. So before any visualization, we would like to have all this information, but just at one taxonomic level. In this case, we would like to have the genus level, a genus level information. So we are going to use the Bracken software that use a statistical method to take a Kraken 2 report as an input and generates this output file that is everything at genus level. So this is okay to make an analysis now. We load this again into a pandas data frame, and before plotting, we are going to take any taxons that are below 1% and assign them the other label. Now I'm going to take this first column of name and this fraction total reads column, and I will generate this fancy horizontal stacked bar plot. And this is it. We are done with the second notebook. Okay, so as we have seen in this webinar, the classification of long reads can be done using these rapid methods that are complementary to the classic approaches. These methods doesn't output too much information. This is okay to analyze next generation sequencing datasets, which are very large. And note that these tools, such as Kraken, they focus on the optimization of the speed and the memory consumption. Again, I should say that the database choice is very important. You have plenty of options available in Kraken 2. It is a good practice to check out what is inside on your database, how often is it updated, check if your species of interest, because maybe they are overrepresented or underrepresented. So you should be aware of all of that before doing any analysis or results interpretation. Remember that these tools are all included in NanoDJ and work fine with the nanopore read data. But you could use them with other technologies. So feel free to import your data into CyVerse and use these notebooks to analyze your data. Also, don't forget to check out our NanoDJ repository for updates. We are going to include very soon these new Kraken 2 notebooks and some other updates. So yes, check out our repo for more information. Thank you all of you. Thank you to the CyVerse people for counting on me for this webinar. I feel very pleased to be here. Sorry for the problems with the slides. And I am here for you for any question that you have. So feel free for ask. Thank you.
Thank you, Hector. Let us see if there are any questions, and there is one that Upendra did answer, but I'll read it out anyway. Somebody asked, will these notebooks be accessible? And Upendra, who is one of CyVerse's science informaticians, answered that these notebooks are already available as part of the NanoDJ Vice app in the Discovery environment. You can find how to access these from the webinar that Upendra gave, and he's included the link there. And I believe, Hector, if you could open your chat window and type in your GitHub repository, we'll make sure that we
Yes.
include that in the wiki page information so that people can check that directly. My question is, what kinds of things are you working on in the future for NanoDJ? You alluded to them, but can you give us a little more of a teaser as to what's coming?
Yes. So at first, we were working on genome assembly. So yes, you would have realized if you have been exploring the app that we have a couple of pipelines for genome assembly, but this was not the same for metagenomics. Now we are focusing on metagenomics, so we are learning new methods and we are evaluating them. So this Kraken 2 notebook is an example of all of that. We are thinking about updating the base calling section because there is a new software that is being used now. And also, we would like to improve the quality control step and include an option to do that like automatically or generate reports with some apps that are already out there. And we are evaluating them because there is plenty of quality control tools. And these are the updates that comes to my mind right now.
Okay, sounds like you got your work cut out for you. Oh.
Yeah, sure.
Okay, if there are no further questions, we can end the webinar, and I'll just remind everyone that in 2 weeks we'll have a webinar on using Git for mere mortals like us who sometimes forget to rename files or to have, you know, things where you've got your versioning crazy or your collaborators are having subsets. So Git is a vastly critical tool for dealing with code. So please join us for that in 2 weeks. And thank you so much, Hector, you and Tamara, and thank you, everybody. See you in a couple weeks. Bye now.